for inviting me here. And yeah, I guess the original plan was that I would be now in Novosibirsk, and we all know what happened this year and the difficulties in traveling and really actually physically visiting. So I hope this way by YouTube and Zoom we can still interact and yeah, share our fascination for science. And I guess I will, as Pavel said, I will talk about sex chromosomes and germline restricted chromosomes, but kind of with a twist towards kind of a long-term fascination that I have for repetitive elements as part of the title here. And that definition might vary a bit throughout the talk, so you'll see that. I'll try to explain this in a couple of seconds. But just to get us all on the same page, what are these strange chromosomes? So I guess just to recap, for sex chromosomes or sex-specific chromosomes, in mammals, for example, that would be then the Y chromosome that's only present in males, and in birds, for example, that would be the W chromosome only present then in females. And then as a second aspect here, germline restricted or tissue-specific chromosomes, I guess the name implies that already. You have some cells of the same body basically that have a certain set of chromosomes, which is shared across all cells, such as also the germline cells here. But then you have an additional chromosome or multiple of those that is called the germline restricted chromosome. So just with that to kind of get us all on the same page, what are these strange chromosomal elements that we're interested in this collaboration, and in general, just trying to understand those tricky bits of the genome. And the species we usually mostly use for our studies are these little birds here, basically. So a bunch of different feathered animals that I think you might have also seen today looking out of the window. The point being, I guess there's many reasons for why we would like to study birds, right? Just this fascinating phenotypic diversity and just amazing speciation rates. So maybe also from that perspective, understanding how the genomes, how the chromosomes have been changing. But if you don't like birds, then another argument could be, well, if you want to understand vertebrate evolution or mammalian genome evolution or reptilian genome evolution, why not start testing methods first on birds? Because they're a bit cheaper to sequence, so to say. This is a bit more like a semi-serious comment here, but the point being, average bird genome size is around 1/3 of a mammalian genome size or half of a reptilian genome size. And at the same time, this is connected also to a lower density in transposable elements. I will explain what transposable elements are in a second, but the point being, all these other genomes are more packed with repetitive elements that, of course, I said I'm very interested in, but for limitations of methods, these are basically not so easy to study at this given point, and we need to test new methods on genomes that are more feasible for doing so. So I would argue also from that perspective, maybe birds are a nice starting point to kind of polish our methods, and then we can go into other systems that are maybe a bit more tricky at this current point. So maybe with these two arguments together, I could convince you that maybe it's interesting to study birds. So then the other aspect, of course, would be, why would we study transposable elements? I have not defined yet what these are. So why would we bother in like the repetitive fraction of the genomes? Why should we look at those points here? And I guess in a nutshell, what transposable elements are is nothing else than a kind of chain letter within a genome. So behaving like a normal gene, having a message to get transcribed, having a message to get translated, but then having a special step here where they can also then lead to making another copy of themselves in the genome. So really kind of a mix of a normal gene with a kind of catch to it so that they can make more copies of themselves in the genome. So for this specific type of transposable element, the, so to say, life cycle looks a bit like this. We have a master element that makes more copies, an RNA level, and then we have a reverse transcription to paste more copies of it throughout the genome. And this is a very successful mechanism. Also, our own human genome is around 50% derived from one another type of transposable elements. And so just to kind of recap that point, just a couple terms of how these transposons look like or how they move around, just to kind of remind you of what we will talk about later in this talk. This, what I just showed you, are the so-called line elements or long interspersed elements. So they have the typical structure like this, encoding their own proteins for doing what they're doing. Then we have also the sign elements. These are very short elements that basically parasitize or hijack the enzymes of line elements, so parasites of a parasite, so to say. One important group I need to mention, because this one will play a role in the first half of this talk, is the group of LTR retrotransposons. And I think most people are rather familiar with this group by the term of retroviruses. So one group of LTR elements are the retroviruses that have very similar structures as this. You have a repeat and a repeat, left and right, this LTR here, and then virus-like proteins in the middle here that are coding regions for virus-like proteins, and then this by a rather large transposable element. And the last group I need to mention are the so-called DNA transposons, or the classical jumping genes, so to say. So these are basically able to cut themselves out through a transposase enzyme that they encode for, and then move around from one place to another. So these are, I think most of you are familiar with the maze transposons that were found decades ago by Barbara McClintock, and basically as a founder of this field. Okay, so I think with these kind of steps here, I want to just then remind everybody also, of course, that genomes are much more than just strings of DNA that have then protein coding genes and regulatory elements, but a lot of other repetitive elements that one way or another compete with each other, or maybe even sometimes go into mutualistic relationships. So these different repeats can be all over the genome, and we can use those to, of course, understand how the genomes have been shaped over the last millions of years when we look back in time. So these can be interspersed, so the same repeat in different spots of the genome. These are the things that I just mentioned, basically, endogenous viruses, endogenous transposable elements. But these can also be tandem repeats. So this is something that I need to mention also, because that might be more relevant for the second half of this talk. This could be, for example, genes that are arranged in tandem, many copies, such as ribosomal RNA genes, or centromere, immune genes. And satellites, for example, would be then in centromeres, or microsatellites and telomeres. So you see a bunch of different terms for all these different repetitive elements, and it does matter to know what these are and where we would expect them when we want to study our genomes and understand how they evolve over time and how they behave, for example, during meiosis. But there's a catch, and this is, I think, the main point of my talk today, to kind of show you what we can do now and what we still cannot do. Ideally, we would want to sequence a chromosome from one end to another, right? Then we get one sequence, and then we can compare that to another species or another individual, and then we can study genetic variation, for example. But this is, of course, not what we do at this point. We have come a long way in the genomics field, but what we still do these days is something akin to a kind of jigsaw puzzle piece. So kind of really using the silly space metaphor here with the picture of the Milky Way, I hope that gets the point across. We have something that's very complex. We chop this into many little pieces, and then we puzzle these things together, of course, not by hand, but bioinformatically. And then we're left with things we can put next to each other, and some things we cannot connect. So by definition, these would be contigs, or every single base pair in that stretch being known as what it is. And sometimes we know then that this contig is next to another contig, so we can call this a scaffold. So kind of, yeah, really like a building scaffold to put things in context together. But as a result of this, what we are left with are gaps of unknowns, of known size, and gaps of unknown size. I'm sorry about that. So the point being, even with many model organisms, there's still more or less gaps in the genomes. And so as a result, we're left with rather short or maybe a bit longer sequences that contain or do not contain these N nucleotides, the undetermined nucleotides. And I think this is something just always good to remind yourselves when we study genomic data. Of course, any study we do based on a genome assembly is only as good as the genome assembly is for certain regions of interest. And so then, of course, it matters how big or small our puzzle pieces are. So it could be, of course, a very complex puzzle for using short reads, 100 base pair reads, for example. So this would be on the order of tens of millions of reads or puzzle pieces for a mammalian or bird genome as well, basically. But it could, of course, be a bit less complex when we go on the order of tens of kilobases now. But that's still, of course, a rather complex puzzle game. So if you take a 1 gigabase genome, we're still talking about tens of thousands, not hundreds of thousands of pieces. And also that's, of course, still complex depending on which regions of the genome we look at. Okay. So I think with these kind of different hints at, I hope I gave you kind of an introduction of the many different reasons of why we study what we study. Maybe one more point to mention here is also, of course, like when we study birds, we have a nice reference point with it, which is the chicken. For culinary reasons or economic reasons, people have spent a lot of money in making very good chicken genome. So we can use this as a reference point of testing methods and knowing what's possible at this point. And I just want to give you this example for when we plot the repetitive element density here across different chromosomes. So you see this, you've seen these terms before, what groups of transposable elements these are. And you see mostly it is around 10%, which is kind of the average of different birds, but you have locally peaks either of gaps or peaks of different types of repetitive elements. So it really depends on what region you're interested in, if it's going to be more or less repetitive and whether you can study it with a given method. And this gets even more extreme on the sex chromosome. So for example, many parts of the Z chromosome are only locally like up to 50% repetitive, so more like a mammal. And so of course that is something maybe more complex to study or more complex to assemble than other regions. And it gets more extreme on the W chromosome, which I will explain in more detail now in a moment. But you see that chromosome is packed full with repeats, and I will explain what these things are. You see this is mostly its purple LTR elements. Excuse me. So I guess the point being, in a cartoonish way, we can say many of these regions are very likely poorly assembled in Illumina assemblies because of the way these assemblies are set up. If there's a very repetitive region, they might be just collapsed or very fragmented at least. And so the idea that bird genomes are stable in those regions is certainly true, but what do we know about those regions in between that are very repetitive and maybe much more rapidly evolving? And this is really the key interest in my group, to really understand those regions that are difficult to assemble and yeah, maybe more feasible at this given time point. And with this, I want to move on to the first study, to actually like move on from the introduction to the actual studies that we did here, being on here now the sex chromosomes or the W chromosome in these little birds here, the birds of paradise. And just one last recap here also. One thing we need to keep in mind when we look at sex chromosomes is that they emerge from a common ancestor. So in this case, proto-sex chromosome pair, in this case, Z and W ancestor that acquired a sex-determining locus. Then if this is linked to a sexually antagonistic locus, so these two are under suppressed recombination, so that either you're a better male or a better female if you inherit this chromosome, then over time, you will basically have more and more degenerated sex chromosomes or differentiated sex chromosomes. So in birds, this means for most species, we have a male recombining kind of normal looking Z chromosome, and then we have a female non-recombining, usually rather small sex chromosome or W chromosome that is very heterochromatic and also usually packed with repeats. And these are, of course, then the things that are of particular interest to us, but also require us to give particular attention to how we assemble these things and how we make sense of the data afterwards. So this is a project that's been led by my PhD student, Valentina, who has been basically kind of using all the methods we had available at this point and combining them, and this way trying to make the best possible assembly at this point. And I would say it's almost comparable to a chicken genome assembly, at least in terms of what some of the regions that we've specifically looked at. And in a nutshell, and I will not explain the methods here, so please ask me this afterwards in the discussion, but in a nutshell, we combined long reads and then a so-called Chicago map for short scale scaffolding and improvement. Then we did base calling correction with short reads. We did further scaffolding with so-called linked reads. And a final step, we got to a chromosome level assembly with a Hi-C map, so 3D chromatin interaction, using that for scaffolding chromosomes or chromosome models, basically. So all these methods together and a lot of manual creation, like really looking at every single gap and every single chromosome model, trying to make sense if this is really supported by the data, lets us think that we have a very good assembly we can now work with. And of course, at this point now, compared to the chicken. So when we started this project, the only other that's of that quality W chromosome assembly was of the chicken. So we would have now a two-way comparison, basically. And I want to just summarize that point by showing you a dot plot. So dot plot meaning if you have one sequence and another sequence, you just put a dot every time there's homology. So in theory, you have two identical sequences would look like this. And let's have a look at how this would look like in the Z chromosome. So here you have our bird of paradise, here you have the chicken. You see all this kind of little background noise, which is derived from the different transpositive elements and other repetitive elements. So this is all the stuff that makes me happy. But of course, this also makes me happy. If you see these diagonal lines here, these are basically collinear and syntenic regions between the two Z chromosomes. So these are single copy orthologous regions where all these different genes are located. You see some rearrangements relative to each other, but overall, we can say pretty much most of these chromosomes, the Z chromosomes, are alignable to each other and look very similar, at least in terms of homology or orthology. So how does it look like on the W chromosome? You can see this already in the title. I say it's low collinearity, but just let's have a look at the data, like visualized this way, the same way. It looks very messy, I guess, long story short. This is something Valentina nicely calls the sea of repeats. And as you can see, the assembled part of the W chromosome in chicken is around 7 megabases, and it's around 19, 20-ish megabases basically in the in the bird of paradise. So it's a bit more like maybe larger euchromatic region here of this chromosome, but overall you see it's just very, very repetitive. So this is the homology from the different LTR retrotransposons that you might have seen on the previous slide, this purple bar. But if you look at this plot long enough, you will actually see that there are some regions that are collinear. So this is here, a tiny thing here, a tiny thing here, a tiny thing here. So just believe me on this. If you stare at this for 10 or 20 minutes, you'll see these as well. The main point being these are the few regions where there are around 24 of genes that are shared between the two species, basically, because there are a few genes left on this chromosome, but everything else seems to be literally a sea of repeats. And as I said before, it really doesn't matter what repeats there are because of certain reasons that I will explain now in the next slides. The first point being, I mentioned before that these repeats are mainly endogenous retroviruses, or a specific group basically of LTR retrotransposons that exist mainly on the W chromosome. And one thing I need to now emphasize also is that these normally come in two different flavors, so to say. When they insert, they are a full-length element. So this is basically definition of a provirus, LTR on the left side, LTR on the right side, and then all the internal protein coding regions. But then stochastically during meiosis, there will be non-allelic homologous recombination because of the homology here. And quite often then you're left behind with so-called solo LTRs in the genome. And so this is quite a fascinating phenomenon. This is well known to occur where you basically in any eukaryotic genome that has these transposable elements. And so our expectation was then, okay, well, if we have an element like this that is theoretically still able to make more copies of itself, and this kind of a dead footprint, so to say, how are these distributed across chromosomes? So what we did then was to order them all by recombination rate, very crudely, of course. Smaller the chromosome, the higher the recombination. Then up here we have the Z chromosome only recombining in males, and the W chromosome not recombining, or at least the region we're looking at. And as you can see here, there's a lot of full-length LTR elements, many, if not most of them, still potentially capable of making more copies of themselves. So they're really potentially proviruses still. So just to kind of put this in perspective, the W that we assembled contains over half of all full-length retroviruses, but is only making up 2% of the genome assembly. So you can really think of this tiny element. It's a tiny chromosome that is kind of carrying all these different potentially active full-length retroviruses or proviruses. And I think that's quite a fascinating point to keep in mind here. What does that mean? Because if we think about it also from the perspective of inheritance, of course, this is only present then in females. And so this is something we then wondered about how to dig into this further. And around the time when we found those results, there was a beautiful preprint published by Doris Bachrach's group. In the meantime, now published in this journal here, a very beautiful study. I can highly recommend looking at this. So what the authors here did in Drosophila, of course, is a very beautiful genetic system for doing so. They made artificial XO males, artificial XXY females, and artificial XYY males. So basically accounting for differences in sex and all possible combinations or dosages of Y chromosomes. So no Y chromosome, one Y and two Ys. And then what they did was to test how long do they survive. And it turned out that the more Ys you have on here, the age of survival and the proportion still alive, you see that if you have two Ys, you die much earlier than if you have one Y, and you live much longer actually when you have 0 Ys. So that's quite an interesting point that led the authors to suggest that maybe there's a so-called toxic Y effect. So basically, also in Drosophila, the Y chromosome is packed full with different transposable elements. So what if just having an extra sex-specific packed, repeat-dense packed region is just posing some problematic effects across the lifespan of these individuals? And I guess, yeah, it's not possible to do this kind of experiment yet in birds, or if it was, it would take a couple decades at this current point. So of course, we can't do this at this point, but what we can do is just analyze the given genome data that's available and phrase some hypotheses based on that. Would that fit with this point? So what happened in the meantime is that we were able to get our hands on a couple additional genomes, either produced by the Vertebrate Genomes Project or also by collaborators. And these are all now comparable to what we produce in the Paradise Crow and of course the chicken genome, which is the best one. So all of these basically different Z and W stages here that we can compare. And long story short, if we just look at how large the assembled W is the very light gray here. This is just like 2%, as I mentioned. It's a super tiny part of the genome. It is usually like this in all the other assemblies as well. So the W seems to be very, very tiny, at least the non-recombining part of the W. And then the same color of gray, if we look at full-length transposable elements here, on this part here, is something between half, if not 2/3 of the genome, except for the EMU, which has much less differentiated W chromosomes. So the point being, wherever we look at, W chromosomes seems to be over-enriched for full-length transposable elements, and almost all of these are these retroviruses that I mentioned before. So this seems to be kind of almost a generalizable point across birds, irrespective of how their sex chromosomes look like, they will have an enrichment of these elements. So then we wondered, can we now kind of formalize this to test hypotheses in the future? And we came up with something that we call the toxicity index. So in this way, we can measure, we can count the excess of intact transposable elements. So this can be any transposons, but in birds in particular, the retroviruses, in the heterogametic sex versus the homogametic sex. So if we expect what the number of these elements in a diploid male versus a diploid female, and then we compare the ratio of that. And so anything around 0 would be no excess, basically the same between males and females. And the further away from 0 we are, the more female bias this would be in this case. So you see the strongest one actually would be here, the hummingbird, and the least strong one would be an emu. And we think this is maybe a nice starting point for in the future testing. Okay, what does this mean in terms of potential toxicity effects on age, on lifespan, and all these other aspects that have been proposed now for Drosophila. And due to limitations of methods, we can't do this in birds yet, but maybe we can keep this in mind for the future. What we can do also at this point now is, of course, with knowledge from where these transcripts come from, we can actually measure if the W chromosomes are expressed or not. I'm showing a lot of different plots here that we did for different transposable elements across different chromosomes. I just want to really just emphasize this point here. W, W, W in chicken, emu, and zebrafinch. The point being, there are, depending on the species, some or many fragments from retroviruses being expressed. Also, that of course doesn't have to mean that they're able of transposition, but the point being, there is certainly candidates that we can study now in the future because they are likely expressed. And so to summarize this point, we think there is a potential for toxicity of W chromosomes because there is expression evidence on the RNA level, but also we find more expression on the protein level in chicken ovaries than testes, the way we had the chance to analyze existing data. So we think all these things together is indirect evidence that a W toxic effect might exist in birds, similar to what was proposed for Drosophila with the Y chromosome. And so to kind of put another perspective of this, so what if you have then a situation where males look like this, rather few full-length transposable elements, and then females look like this? You have this W just kind of keeps on radiating transposons or retroviruses all over the place. What if this, for example, then really kind of explains why females in birds tend to live shorter than males, basically? So this is really almost a general phenomenon across birds. Many species, if not most species, are having this pattern. What if the content, such as retroviruses on the W, plays a role in that? And keeping in mind what I showed you before with this sea of repeats comparing the chicken and the bird of paradise, if this is true that W diverges very rapidly, could this, for example, then also contribute to Haldane's rule? So the Haldane's rule meaning that if you have hybrids between two different species, in this case of birds, the one that is infertile or has reduced fertility will be the female, because maybe then the W chromosome is causing problems because of lack of repressors of these elements, etc. So a bunch of things, of course, we cannot test at this point, but we're thinking about the next steps to really test both on a cytogenetic and a genomic level what this means basically for the organisms, and what does this mean also for bird speciation in general. And yeah, with that, I would like to summarize this part of my talk, the first part basically, and I hope I could convince you that we can now, first of all, we can now build nice enough genomes to compare the W chromosomes between species. Firstly, I showed you bird of paradise and chicken comparisons. So quite rapid and massive structural changes across bird evolution, mostly because these chromosomes have been bombarded by retroviruses, and most of these are still at full length, with the effects that this is very likely some kind of toxic effect, such as maybe a female biased mutational load due to during the lifespan basically of the organism, but maybe also in terms of like mutational impact across germline development. So all these things are of course still speculative at this point, but something worth testing out in the future, right? So if we have the chance to look, for example, at to compare basically spermatogenesis and oogenesis and look at expression of different retroviruses in those stages. Okay, so that's all for sex-specific chromosomes or W chromosomes. I would like now to shift gears and talk about this little bird, the zebrafinch, and more specifically about these germline restricted chromosomes that I mentioned before. I also think this is very interesting to especially Pavel and Anya here in the audience, but in general, I mean, this is the reason why we started collaborating in the first place. And I would like to kind of take you now on a slight journey and explain how we got into this, because I think with all the stuff that we found when we did this project, I basically didn't expect almost any of these results. I had almost the opposite predictions almost every time, and never have I been that happy to be wrong basically with my predictions, because it turns out to be such a fascinating topic. But let's take a step back. So how did this start in the first place? This has been actually known for 22 years now, this chromosome. There's a beautiful study in 1998 showing that looking at meiotic chromosomes of zebrafinches, they found a very strange element. So when you just compare now somatic karyotypes here, males versus females, this is just the 8 largest chromosome pairs. You see the main difference is just the W chromosome. So all beautiful, this is what we like. And then when you look at germline cells, in this case from the same male bird, and you just look at the largest chromosomes, you find this extra massive, in this case, lonely chromosome that is larger than this chromosome here. This has been called chromosome 2, which is 150 megabases in size. So this should be something on the order of like 160, 170 megabases. So massive chromosome, over 10% of the genome size. So what's going on there? And yeah, this is something I was kind of, I guess, loosely aware of over the last years. Just kind of, I guess, like many of us, just thinking this is some odd thing. Okay, maybe zebrafinches are just strange. But because this is such a massive chromosome, there's been actually beautiful work over the years, nicely summarized in this paper here, to really follow the behavior of this chromosome, I guess, across male and female meiosis. And so to very much simplify and summarize, what you're left with is, at the end of male meiosis, sperm cells without a GRC, because it's thrown out during spermatogenesis, and in females, you have a single GRC. And then you have a black box here of embryogenesis, which is at this point unknown. And then when you're adult, you have never ever in the soma the GRC, neither in males or females, but in males, you have 1 GRC in the germline, and in females, you have 2 GRCs in the germline. So you see a lot of black boxes, a lot of question marks, but overall, this is kind of the main points to keep in mind about the behavior of these chromosomes. And it's very fascinating from that perspective, right? Why would these birds have just an extra chromosome in one tissue type, but not in others? And this is one important key point to keep in mind. So because it's such a big chromosome, it is actually possible to observe it in cytogenetics and like make these very beautiful studies. But in terms of genetics, when we started this project, we started this now almost 4 years ago. Yeah, pretty much 4 years ago. In terms of genetics, actually, there was very little known. So there was, at this point when we started this, there was just one study, this study here, that had sequenced painstakingly. This is before the next-gen sequencing revolution, 18 kilobases. So this is very impressive, but it's, of course, a tiny fraction of this chromosome if we look at this from now. And what they hypothesized here is that what if this region, at least, is just an extra copy? So it's kind of something that is present on a regular chromosome, the A chromosomes, so to say, that's just been extra copied onto the germline restricted chromosome, kind of almost like an aneuploidy or a trisomy. You have an extra copy of that. But it gets even more complicated than this, because this region is present in a single copy in the regular chromosomes that they sequence here. 27L4 is the region, name of this region. So this is what you expect in a diploid organism. You just have these 2 chromosomes that are lighting up. This is the chromosome pair that has a single copy of this region. But when they then stained the region in germline cells here, meiotic cells, sorry, you see there's a bit more than these 4 dots that you can see on this side here. So this led the authors to conclude that it's not just copying over a region like a paralog onto this GRC, but actually then expanding that in copy number. So into many copies. We tried at some point to sit down with a glass of wine, and we got to something like 40 copies, but it's a bit hard to say, right? I mean, you can just squint your eyes, and it depends a bit on how good the the picture is resolved here on the projector. But the point being, long story short, we assume something is basically behaving like an extra copy, so like an aneuploidy almost. But in addition to this, it will have many, many similar copies, right? Otherwise, you would not get the fish signal here. And so these are something, of course, to keep in mind with this puzzle game that I mentioned in the beginning of my talk, because when we want to now study this on a genetic or genomic level, we need to be aware of those limitations here. And it basically happened to be a very lucky coincidence that when we started this project, our sequencing center was testing a technology called 10x Genomics Chromium. This is a linked read sequencing technology. Unfortunately, this is kind of dead now. It was discontinued, but it's a very promising technology, and I'm very happy to give you more explanations later on also why that is. Just want to also mention this was a project led by my first master student, Cormac, and by a postdoc in my group, Francisco Paco. And so basically, they were looking at the data that we got from this. But I need to just first also recap what this is basically. This is basically still short read sequencing, but a very fancy way of doing short read sequencing, because it uses a microfluidics device where the DNA, each DNA molecule, roughly each DNA molecule, meets a separate little bead here that contains different adapters and different barcodes to be able to trace back all the short reads from that specific barcode afterwards. So you have separate oil droplets, and each of them you basically make a tiny little library preparation, and then you sequence everything together afterwards. So because of this, we're able to then link the reads together. That's why it's called linked read sequencing. And when dealing with this data, we then basically developed some new methods to really disentangle where these regions come from. The assumption here being, we have a very nice reference genome for zebrafinch, and reference genomes are usually just haploid, basically, just the two chromosomes collapse together. So here we just have one A. So now when we have our linked read data from the soma, we will have two haplotypes here separately. And of course, in the germline, we expect the same two haplotypes, because this is the shared A chromosomes across all cells, of course, plus at least one additional, if not more, haplotype that will be then existing only in this sample versus this one. So this is a subtractive approach, but also mapping approach that we developed there that allows us to find these regions either by SNPs or by coverage. And if we have SNPs, for example, like the single nucleotide variants here, we can then also trace them in a transcriptome level, because they will only exist then in reads from here, not here, and then similar level also in proteomics data, basically. So these are the different steps that we analyzed also in our first study to really test those methods, and just as a proof of concept, show that we can actually visualize and find those regions. But yeah, long story short, I think the main point or main reason why we studied this chromosome had nothing to do actually with the chromosome itself, but more the repeats that I was expecting on this. I have to say that this was around the time when we were finding lots of retroviruses on the W chromosome. So I was very curious just to see, okay, how does the germline restricted chromosome look like in retroviruses? Does it also kind of act like a sponge for all these retroviruses? And what does this mean? Yeah, and how wrong was I? Well, it turns out there is no enrichment in repetitive element on this chromosome, at least with that definition that I mentioned before. So it was very strange to get that result. I think I was scratching my head for a while. We even sequenced 2 additional birds from another population with a different method, just to make sure this is not an artifact from the methods. But long story short, if we look at short tandem repeats, for example, here counting in soma versus counting in germline, everything kind of follows a diagonal line, right? So there's no enrichment one way or another, as one would expect if there was something highly repetitive just on the GRC, basically. In a similar way, we can also analyze here the so-called repeat landscapes. This would be basically using transposable elements and satellites, larger satellites, plotting them here on this axis being the relative age, so to say, and here the delta between the proportion in one tissue, testis, minus the proportion in muscle. So the point being, if something is enriched on the GRC, it will be positive values. If it's under enriched, impoverished on the GRC, it's negative. And as you can see, except for a few in green here now, LTRs, everything else actually is negative. So actually, overall, it's not just that there's no enrichment in repetitive elements on the GRC of zebrafinch, but it's actually fewer repeats overall than the rest of the genome. And so, yeah, I was completely wrong basically with my prediction here. And yeah, what does that mean? What is going on with this chromosome instead? Especially when I just showed you that the previous work from around 10 years ago showed there was something else repetitive. So what's going on there? I need to use a slightly different plot now. This is the so-called Manhattan plot. here we plot all the chromosomes, the regular chromosomes, the A chromosomes, next to each other. And on this axis here, we show now the ratio between sequencing coverage in one tissue versus the other. The assumption being, of course, if a region only exists on the regular chromosomes, the A chromosomes, the coverage should be plus minus the same between one tissue and the other, right? That's the expectation. Plus minus, I guess, meaning a bit of just stochastic variation. So most of that stuff here, I guess, is just, this is just kind of background noise here. Keep in mind the log2 scale. But then you see these local peaks, and some of them are rather long peaks, others are shorter peaks, but overall, across the different chromosomes, we have basically peaks of coverage. So this means a region that is present just in a single copy on the regular chromosomes, such as in soma then, is present in many, many similar copies in the germline tissue. So that means the GRC very likely contains just extra copies, not just one, but many extra copies. We did the calculations. I think for this DPH6 gene, we get to a coverage of something like 300 copies or so. So the point being, the GRC is not really repetitive from a classical repetitive perspective, but it's repetitive in terms of having acquired regions that used to be not repetitive, if that makes sense, the wording here. So you start with single copy regions, you put them over, and then suddenly you amplify them in copy number. And of course, then we can use that also to visualize this cytogenetically. This name you've seen before, this was actually checked cytogenetically 10 years ago as being a point here. And this is the highest peak that we found in our data, basically, and we then also tested that in fish, and we literally also found a very nice blob here of signal, basically in spermatogonia cells here. So the point being, we can visualize these things now, and so we think we have an idea of what is on this chromosome. But I want to now use the next slides just to show you what we think is on these chromosomes without speculating too much. Just kind of show you the data, and then we can maybe use the discussion to speculate what this means. So the first point being, we can also then test the expression of these different GRC copies, basically, because of the sequence variants that we sometimes find. So this is just one way to visualize this. We have one tissue, everything being mapped against the reference of this gene. That's why it's in gray. And now just keep in mind, this is the same bird, just a different tissue. This is the testes from the same bird. We have, again, these gray reads that are mapping, that are identical to the reference. So these are achromosomal paralogs. And then we have some reads that always have multiple different colors, each of the colors being a SNP, a single nucleotide variant. So all these different reads here basically are then very likely from the paralog on the GRC. So we have a means to really, in this case, visualize for you that these copies are not just existing, but we can trace them on the DNA level. And for example, when we look at expression data, here we have all these gray reads again. These are then the transcripts from the regular copies, the achromosomal copies, and then these other reads here, which are then transcripts from the GRC paralog very likely. So just a very brief recap of how this data looks like. But to summarize that, we have at this point now evidence for expression of something like 32 genes or GRC-linked paralogs in the ovary and 6 GRC-linked paralogs in testes. And on a protein mass spectrometry level, we're even able to also find protein expression with the specific peptide, so the specific amino acid changes for the GRC in testes and ovary. Then another thing I didn't mention yet is, okay, what are these genes then? So there are overall 115 genes that we find at this point with high confidence. There's a couple hundreds more that we think exist. But the point being, so some of these, as I just showed you, are expressed on the RNA and protein level, but overall also it's not a random set of genes. This is why I put this plot here. This is a gene ontology enrichment analysis. You see the strongest enrichment is in terms of female gonad development. And I will not speculate about what this means, but the point just being, this chromosome contains multi-copy genes that are expressed and for some reason enriched in specific gene ontology terms. Yeah, and then another thing I also need to mention is, well, at least some of these genes are then also potentially under long-term selection. So this is an analysis we did for genes where we have enough single nucleotide differences between the two paralogs, because right, if we have specific variants between the achromosomal paralog and the GRC paralog, we can then use that to see, okay, are these non-synonymous changes or are these synonymous changes? This is a typical DNDS analysis that is used for testing for signatures of long-term selection. So we were able to do this for 17 genes that have enough single nucleotide variants to have the power to do so. And from these 17 genes, 9 appear to be under long-term purifying selection, and that includes a couple here where we also have expression evidence. So they're expressed and they're under long-term selection, and one of these genes is under positive selection. So again, I'll keep the speculation of what this means or what it does, the GRC, to the discussion, because I would say at this point we don't know if we really have found all the genes. I mentioned this is just a subset of the genes that we find with different lines of evidence. So it could of course be different genes that are actually playing a role here in whatever the GRC is doing. But I want to just now emphasize another key point, which I think is just something very interesting to keep in mind. When we have sequences of the different paralogs, the GRC paralogs and the achromosomal paralogs, and they have sequence differences between the paralogs, we can of course make a phylogeny of those and then see what happens here. So here, for example, we have now zebrafinch up here. This is the achromosomal paralog. And down here we have zebrafinch, the GRC paralog, and all these other species are somatic genome assemblies that are available in databases. So you see here, according to this, that would be a gene that very likely diverged since the common ancestor of all these different songbirds sampled here. So I had a hard time believing this when we first made this tree, and that was a couple of years ago when basically exactly I think in that week or that same month, basically Anya and Pavel put their beautiful cytogenetic study on bioRxiv. So I actually have to say this was kind of the deciding factor for me to believe this result, because you basically showed with very complementary methods that everywhere you look, whenever you look at a songbird and you look at the germline, you will find a GRC. So that's super fascinating, and basically very nicely like complementary evidence now that very likely most, if not all, songbirds have this GRC. So this is, I think that's super exciting, but of course, such a strange thing, like why did nobody notice this before? Our two groups have been looking at this, right? I also want to mention there's some genes where if you look at the same topology, so this is the same topology as on the left side, the same species, everything else is the same, except for the position of this red GRC paralog. Now it's up here. So you see these are clustering together, whereas the sister subspecies, this is the Timor zebrafinch, is clustering at the outgroup of these two. So this would be actually a gene very likely that has arrived on the GRC kind of recently. This is a subspecies specific gene, whereas this one is a very ancient gene. So there's a whole range of old versus young genes that I can summarize as this. So this would be the species tree of the species I just showed you. And keeping in mind, of course, we have for this study only sequenced this zebrafinch here. We didn't have any germline information for that. So this is what we're working on right now to kind of validate that, of course. But the point being, just from one's perspective here, making phylogenies, we get some old genes, some intermediate age genes, and a lot of actually very young genes. So this is super fascinating. Does this mean it's a very fast evolving chromosome? We don't know yet, but we suspect so. And another point I need to mention, where do these genes come from? So literally all of them have at this point a paralog on a regular chromosome. So when we have a paralog, we can of course then make a line from where they're sitting on the GRC. We don't know how this is arranged yet. We're working on the assembly. So this is just a placeholder, and then we draw a line to where they come from, where the other paralog is located on the other chromosomes. And you see they're literally from all over the genome, basically. Large chromosomes, intermediate chromosomes, and microchromosomes, and even the sex chromosomes. So they're almost like a sponge picking up genes from all over the place, which I personally find very fascinating. And I'm still struggling to explain that mechanistically. How do they arrive there? And we hope we can fix this now with more genome assemblies and better assemblies in the next years to really understand that mechanistically. But I think the last point I want to mention here for this part of the talk is just to say we have a hypothesis of when exactly this chromosome emerged, because we have one gene which I didn't show yet. This is called TRIM71, where if we make a phylogeny, these are basically oocynes. There's the true songbirds where also Pavel and Anya have been basically looking at so far. I don't think you had access to subocene cytogenetic data, and also so neither did we, but we found these different assemblies and databases from somatic tissues. And so if we make a tree of that, you see that the GRC paralog is clustering outside of that. So if that's true, and we hope to be able to test that now, this would mean that this GRC emerged actually in the common ancestor of all passeriformes. So that would be actually not just 5,000 species of birds, but maybe closer to 6,500 species of birds, which is rather fascinating, right? So to summarize this part of my talk, I guess I hope I could convince you that the GRC is repetitive in a very special sense. You have just single copy genes that now became multi-copy on this chromosome for whatever reason it is. They're also very likely ancient and at the same time very rapidly evolving, whatever that is, or whatever the reason is for that, basically. And because we have now some limited evidence for some chromosomes being expressed and also conserved, we suspect it must be doing something. And there's a bunch of things that I'm putting here, and I also put here insert argument. At this point, I think we just need to speculate and make a bunch of different hypotheses that we can test, because it is very unclear what this chromosome might actually be doing. And I just want to now finish with 2 slides to kind of give you some sneak peek of what we have found in the meantime. This is all unpublished data, that's why I put this here. So feel free to talk about it. I just want to say that I put this here because the numbers might change, because we're still testing these things. One example is, I said the GRC of zebrafinch is not enriched in transposable elements. Well, the first species we looked at now is this little blue bird, same family, Estrelidae, 10-50 million years diverged. And here we actually find an enrichment of one very specific type of LTR retrotransposons of retroviruses. A lot of them, actually. This is work done by Jesper recently. So what does this mean? Is the zebrafinch the strange one, or is this one the strange one? We have to understand this further, but it looks more complex. Now, when we look at this, in terms of genes, it still holds that there's only few genes that are actually shared or ancient genes, and many of them, in this case, actually, this little blue guy has more unique genes, basically, than the zebrafinch. So also from that perspective, the patterns are starting to look more and more complex. I should also mention the blue one here has not as many high copy number genes as the zebrafinch. So this one has fewer genes with high copies. This one has more genes with lower copies. So who's the odd one now? We don't know yet. We need to fix this out. And the last point I need to mention also is, for some of the genes where we could get decent assemblies, it's very few yet. We're still working on improving assemblies, but for some of them where we could get decent assemblies, it looks like almost always the introns of the GRC paralogs are significantly shorter than from the achromosomal paralog. So this is a very fascinating thing. One could speculate that this reflects some kind of selective pressure, but what that would mean, of course, needs to be elucidated. But the point here being, for example, here in red, you see this is a paralog from the GRC mapped against the reference genome. And wherever you have this huge gap here, this is basically a deletion on the GRC relative to the reference. So this is not loss of introns, but independent deletion of one big intron here, one big intron here. Here's even deletion of an intron plus one exon. So the story is looking more and more complicated, and I'm curious to see what kind of questions or also speculation you have in the discussion, which we will be able to start very soon, because I just want to kind of leave you with a lot of names. Thank everybody on the slide here. I put the main actors of the respective projects on the slides because there's just too many names here. I want to thank all of you for inviting me here, my group for being amazing during this pandemic, and of course, Vertebrate Genomes Project and others for providing data, and lots of funding agencies for providing money to do these things. And yeah, I'm very happy to take questions. Thank you so much.
Excellent. Thank you very much, Alex. Thank you very much. Fascinating talk. So, well, the first question, I will ask the first question. So about the GRC, what do you think? It looked like it occurred, well, like 40 million years ago as a vehicle. And now anybody who wants just enter this vehicle. Is it true?
I would suspect so. I think that's a very good point. So I think that's reflected just by the amount of genes from or regions from different chromosomes. So I guess what's the oldest one? I think the oldest one is from, forgot which chromosome it was, but I think it was either chromosome 2 or chromosome 5. I don't remember right now. But the point being, we think the oldest genes basically are just from 1 or 2 chromosomes. We have to, of course, dig deeper in those also to make sure which one was first. But yeah, it sounds like this started as a vehicle, maybe like a B chromosome, so kind of supernumerary polymorphic chromosome. Why and how it got fixed in the population and how it got eliminated is a different story. But then afterwards, just genes keep on accumulating. Exactly.
Yeah, but vehicle is the same. Yeah, it's amazing.
Exactly.
Because what Anya found that in different species, the GRC contains big clusters from different chromosomes. For example, in zebrafinch it's 3rd chromosome, in marten it's say 6th chromosome, and like this. Okay, exactly. So anyone have questions? Just raise your hand. While you are raising your hand, I will ask my questions. Okay, from the end, what gene shows the positive selection?
Let me go back to that. I was just one. I think it's called PUF something. The thing is, I need to PUF 60 exactly. So there's the thing I need to mention is here. What we're looking at is really a subset of genes that we can analyze just with one species. So keep in mind we need to be able to find those orthologs and paralogs in the other species as well, for which we didn't have germline information back then. So this is really just a subset, maybe the most conserved subset of genes that we could study here. So there's just one gene under positive selection with that. So I would suspect we will find more genes once we sample more species and also have better assemblies for those. And at the same time, I would kind of wait with my speculations of what this means until we really have a complete, like an exhaustive list of genes, because at this point, it is really just a subset of genes. But I guess overall, what many, not most of them, but many of these have in common is at least some of them, especially these two oldest genes here, BIG1 and TRIM71, are both involved in cell differentiation and early embryo development of mouse. So if that necessarily means that this was the first reason of why the GRC emerged or got fixed in the population is a different story, of course. This is something we need to really be able to test by saying we have looked at all the genes, which we haven't at this point, but it's a starting point.
Okay, Denise Larkin has raised a hand. Can you talk, Denise?
Yes, I can. Can you hear me?
Yes.
Well, I'm somewhere in the forest, so yeah, you might have some issues.
Any birds around?
Yes, we have quite a lot of parrots here in St. Albans. Yeah.
Shoot them.
Well, I will not today. Yeah, so I have a question about density of transposable elements on a GRC. So you showed on your slides that you have quite a lot of recent transposable elements and low density of ancient transposable elements. Does it just mean that you start accumulating those elements which were active after this chromosome existed in a genome? So I think it's not this slide, it's earlier slide where you show underrepresentation of some transposable elements and overrepresentations of others. When we looked at this with Anya and Pavel, my impression was that you just get those which were active after this chromosome was in a cell. And well, those ancient elements which were present, well, active before formation of songbirds, they actually didn't have a chance to accumulate because they were not active. And the only copies you have are those which were copied from the genome.
Exactly.
Yeah.
Thanks, Dennis. That's a very good point. I think it's reflecting really the kind of the age of the germline chromosome from that perspective. But maybe another point I should make here in this context is that if what we see now with these old paralogs of the old genes is true, that they have a lot of deletions in introns, etc., it's also possible that for some reason there's a higher turnover either of genes or in general just a high deletion rate of regions on the GRC that are not constrained. So what if the older transposons have actually also been deleted over time? So this might also kind of reflect a mix of that, both like a limitation for what can accumulate and when, and at the same time for how long they stick around on the chromosome because they get deleted at some point, basically. At least maybe faster than the rest of the chromosomes, the A chromosomes.
Well, we looked at similar stuff in mammalian breakpoint regions in chromosomes, and of course you will find a lot of recently active transposable elements in those regions because that's open chromatin areas. But you will see underrepresentation of ancient elements, not because you delete them, but mostly because you insert novel transposable elements and you destroy old ones. Exactly. So could it be a similar mechanism here that you just insert new elements and you destroy old ones? And something else for you to look at is when you speak about genes with shorter introns, is it possible? Yeah, I understand that you kind of say that it could be deletion of certain regions, including exonic sequences, but could it be that when those genes were inserted in GRC, they were shorter just because you had less transposable elements in the host genome? So you kind of get relict copies of those genes in GRC.
That's a very good point. So as I've looked at a couple of these things manually so far. This is just a handful of genes, but as far as I can see for these ones, there are these introns here on the regular chromosomes, the A chromosomes, contain almost no transposable elements. So also there, it's not, this transposon jumping alone, I think, would not explain the difference in the intron length. So it looks more actually that this is just deletions unique to the GRC. We can actually validate that by aligning the introns with other species that are available. So for the A chromosomes, and there it also looks like the introns are more or less similar size, similar length, similar content, very few transposons. And then it's always the GRC that's different, the one that has basically kind of almost secondary deletions of that. So I would suspect at least that for those limited genes that I've looked at, that's likely not the case, that differential transposon accumulation is a point there. But I would also say, I mean, we need to look at this more exhaustively, and also the first point also being we need to make better assemblies, because right now they are very fragmented, and so we might be only looking at a subset of very easy genes, so to say.
All right.
Thank you.
All right.
Thank you, Dennis.
Minya Fishman.
Hi, can you hear me?
Yeah.
Great. So first of all, thanks a lot for a very interesting talk. And the question I have is actually, you said that you haven't found a lot of repetitive elements on the GRC, but could it be that they are GRC specific and you don't have them in genome assembly? And the related question is whether you can somehow, for example, from the portion of unmapped reads, estimate how well you covered the GRC.
Thanks, Minya. These are two very good questions. So to answer the first question, so we thought this would be maybe just assembly issues. So what we do here, actually, I should have mentioned that what we do here, actually, this is the proportion of reads. So this is really just, should be independent of assembly issues. It is really a pattern we see both in the assemblies as well as in the reads. And to your second question in terms of how much of the GRC we cover, the difficulty, I think, is we have rather decent assemblies for the older GRC regions, because they have enough SNPs to differentiate them. But it looks like most of those genes that are very more young genes on the GRC with very few differences, maybe 1 or 2 SNPs, etc. Many of them are actually very poorly assembled, or sometimes kind of almost like a chimera with the A paralogs. I've looked at some of them manually just to kind of see where the SNPs would lie in the assembly, and it is very messy. So the thing is, keeping in mind these linked read methods are basically optimized for making a diploid assembly. So you assume everything is roughly there in 2 haplotypes. So I guess one of the limitations there would be just if you have a third haplotype like an aneuploid that's very similar, by chance you will just have a mix or chimera or collapsing of that third haplotype. So what we're doing right now is actually to try to optimize that by developing so-called barcode enrichment method. So this way we try to enrich either for the GRC haplotype, or we remove the A haplotypes, and this way we reduce the complexity of the data. So this is what we're working on right now, and it looks like we've been actually missing quite a lot of regions. So that's also why I was saying like the point of having 115 genes is very likely a significant underestimation, because we're only now like really able to make better genome assemblies and find more and more genes there. So I would say it's maybe at least twice as much, if not more, in terms of genes on the GRC.
Okay, thank you. The question from Vladimir Trifonov.
Hello, can you hear me?
Perfect.
Can you hear me?
Yes, yeah. All right, go.
Thank you. Thank you very much for your talk, Alexander. And I've got the following question. It's about the toxic effect of the W chromosome. So the question is, if other elements of the genome that contains these huge heterochromatic blocks and might be enriched in retroviruses could be also possibly toxic. And this effect of females in birds, that this shorter lifespan, can it be connected just with the fact of the dosage of the Z chromosome, which is reduced twice, which contains so many essential genes and present in females only in single copy? And how do you explain the fact that Z autosomal translocation are so rare in birds? As far as I know, there were only 2 reports and 1 article in preparation about this. Okay, thank you very much.
Thanks so much, Vladimir. So there was 2.5 questions, almost 3 questions. I tried to keep them and try to answer them in the order. So the first question was related to whether there are other regions in the genome, like the autosomes, for example, that contain full-length LTR elements, for example, or other elements. The thing is, of course, I would still suspect we're missing many of these very heterochromatic regions on any chromosome, irrespective of sex chromosomes or GRCs or autosomes. So I would not exclude the possibility that there are regions that are also toxic there. So what I do wonder though is then in this context, maybe the toxicity is not itself the presence of these elements that are capable of transposition, but just the fact that there's so many of them in females. Because if you think about it this way, also many mechanisms of silencing transposons and endogenous retroviruses are either very sequence specific or also very dosage specific. So from that perspective, what if that kind of poses an extra obstacle or hurdle once there are other environmental stressors, for example, or just stochastic effects during aging that just mean just because having to deal with 3 times as many nasty transposons that are able to jump, more accidents can happen than in the males that have much fewer than those. But at the same time, yes, I would not exclude that we have a lot of full-length elements also in the unassembled regions on the regular chromosomes. So that's a very good point. The second question was related to the, what was that? I think it was the dosage of the Z chromosomes, right? Yeah, exactly. So I think this has been discussed before, that maybe that's all just reflecting the presence or absence of the second paralog, so the gametolog on the W chromosome, because the bird Ws also do differ quite a bit in their gene content. I do think these both points play a role there, but I guess it's kind of hard to exclude one or the other at this point without experimental evidence for what is exactly going on. So I mean, this W toxicity versus Z toxicity or dosage toxicity there, I think it's just maybe good to keep both of these points in mind for future experimental designs, because supposedly, at least, birds have means of gene by gene dosage compensation that should kind of deal with those aspects as well. At least this has been proposed before, but of course, what that means in terms of deleterious effects during aging, etc., is something I'm not aware of, and I guess has to be looked into further. And I think the third question was?
It was about the translocation with autosomes,
yes.
why they're so rare in birds.
Thanks, Vladimir. Yes. So I think this is a general thing that has been kind of attributed on the one hand side to this kind of stability of bird karyotypes. So it seems to be just in general that there are very few translocations, not just from Z to A, but in general between A chromosomes or any kind of chromosomes actually between birds. I mean, one suspicion there is just maybe compared to mammals, for example, if you have lower repeat content, that would also just maybe provide less or fewer substrates for non-allelic homologous recombination, and just this way, just being able to translocate something between chromosomes. Another point I need to make also is that unlike mammals, where you have line 1 retrotransposons that are basically every now and then they just grab a transcript from a normal gene and then make a so-called retrocopy, like a reverse transcript copy randomly in the genome. These transposons don't exist, so we don't have any equivalent in birds that would be able to make these so-called retrocopies or retro pseudogenes. So I think the average mammal has a couple thousand of those retrogenes, if not 10,000, if I remember correctly, versus birds having less than a handful, I think. So from that perspective, I guess the mechanisms of moving things from the Z to the autosomes, but also similarly from the A chromosomes to the GRC, those mechanisms I think are, yeah, a bit limited, I guess, as far as I can say right now. I'd be curious to see if anyone has a suggestion of what could be playing a role instead if we don't have retrogenes and if we have fewer translocations happening by chance anyway. So very good points.
Okay, thank you. Well, just to the same story about the W toxicity. Do you make any sense of that? You know, in ostrich you have very low ratio, in a hummingbird very high. Does it make any sense about the weight or speed of flight or whatever?
That's a very good question. I think this is also one of those mysteries. Why are birds so different in that regard? I can just point out the one point here.
That one. Yeah.
So I should go this back. So this is not distracting. So, I mean, the one thing is, okay, yes, you have the flightless ratites down here, and most of these ratites have rather so-called homomorphic sex chromosomes. So the W is recombining across most of the length with the Z chromosome. So what we then analyze is just a tiny fraction that is then female specific, so to say. So I would say naively, I guess, I mean, what they have in common is, of course, that they're flightless, but at the same time, I mean, this could be just reflecting their close relationships, the phylogenetic differences there. So the general pattern being that across the Palaeognathia or the ratites here, you have just longer recombining region here. If that necessarily then explains that you have the lowest toxicity here, or if it's related to another life history trait, of course, is something we can't say at this point. I mean, also keeping in mind, especially these retroviruses, I mean, we call them endogenous retrovirus because they're inherited through the germline, but new lineages can come in from the outside every now and then. This is so-called exogenous retroviruses. So if they infiltrate from the outside and get into the germline, then they can become a new endogenous retrovirus lineage. And so it is very well possible that there's some other life history traits, such as ability to fly or effective population size, for example, or also the habitat or distribution area that might make you more prone or less prone to getting new viruses coming in, especially because I said most of these things here, toxicity aspects here, are related to this endogenous retroviruses. So that's a very good point, Pavel. Yeah.
Or just phylogenetic signal, you know, the tree is not very dense here. Yeah.
Exactly.
Yeah. Okay, Nikolai Borisovich,
Yeah, okay. Thank you very much. Very interesting. You see, we can list a lot of examples of chromosome regions with the features similar to the features of Jersey. For example, cluster of duplicons in mammals, mainly mammals, or B chromosomes. Yeah, they also have some regions from different chromosome regions of normal chromosomes. For example, heterochromatic regions in pericentromeric regions especially can be transcribed and very intensively transcribed in some stage of ontogenesis. We can remember about HSRs in some cells. Okay, it's a little different story, maybe it's oncology, but all of them are in the somatic cells, no problem for elimination. Why it's necessary to involve such very strange mechanism for elimination of Jersey? Why it's not impossible, but it's not possible to have a Jersey just in all cells of the organisms?
Exactly. Yeah, it's a very good point, Nikolai, and thanks for pointing out also the similarities with other systems. Especially, I agree, fully agree. I find the similarities like with B chromosomes very striking, that they seem to also accumulate regions from all of the chromosomes when people start sequencing those. So that is very fascinating. I'm going to wave my hands now to kind of say this is speculation time. So basically, really, I have no data. I've just kind of some ideas, and I showed you before. I was very wrong with many of my other predictions for the GRC project. So I could be completely wrong now as well. So just absolutely honest, your speculation time.
We very often are wrong. It's no problem.
Exactly. So the thing is, I think that what we're speculating right now is, okay, maybe what if the GRC started as a normal B chromosome? So at some point, the common ancestor of Passeriformis, it was basically, polymorphic, and maybe it was, every now and then you had an organism that had it in every cell. Sometimes you didn't have it at all, multiple copies, etc. And then now it becomes very non-parsimonious. Now you have to invoke several steps. Somehow then some genes arrived on the GRC that were very beneficial, especially in the early germline development, and then that was very good. But at the same time, if you kept these around too long in other tissue types, this would then be, of course, deleterious. This is something that's called antagonistic pleiotropy. So you have the same gene that is beneficial in one context and deleterious in another context. So, and the thing is really emphasizing, this is completely speculative. You can go to the slide actually to point that out. It is just really based on this very incomplete data set that we have at this point. And this is why I save the speculation for now. The point being, I think it's this one here. The point being, the two oldest genes here are both involved in I think it's pluripotency during early embryo development, basically. So this is both known from mouse systems. And so the point being, what if somehow and again, still speculation, these genes seem to be always in single copy in vertebrates as far as I'm aware of. So what if somehow having more copies is actually beneficial for the early embryo development, because it actually allows it to develop even faster? Again, I'm still speculating. So what if having higher gene doses at certain stages are actually beneficial? But if you keep this around in somatic cells, you have a higher chance, for example, to overexpress them over your lifetime and develop cancers, for example, as one speculative aspect there. So what if then, and this is where it becomes non-parsimonious, what if basically you have something like this, you have a beneficial B chromosome polymorphic in the population, so it sweeps to fixation because of that, but at the same time you have the deleterious effects whenever you keep it around in the soma. So as soon as somehow, and also there, I have no idea how right now that works, how you remove this chromosome mechanistically, but if you find a way to remove it mechanistically, some epigenetic marks, or you have a specific centromere structure in there, or some other sequence-specific mechanisms there, what if then you reduce that deleterious side effect basically of having this chromosome? And so there's a transition phase where we would have to assume you go from a B chromosome to a fixed B to then a soma eliminated B chromosome, or by definition then a GRC. And what if that was so successful back then in the meantime, as Pavel said earlier, like it just became a vehicle for other things as well. Suddenly you have a vehicle that is both has a male-female dosage difference, like one in males versus 2 in females, but also has this germline soma presence absence pattern, and then that can be used for many other avenues. I think, for example, many of these genes down here are then more involved either in male or female gonad development, etc. But again, very speculative, waving my hands again, because this is really just a subset of the genes that we can analyze most easily in our proof of concept study. So it's possible that the actually interesting genes that are responsible for what's going on are not studied yet because they're too divergent, or we couldn't assemble them yet, etc. So I could be completely wrong with what I say right now. So, and I would be also happy to be wrong as long as we can prove at some point what is going on here, basically.
Well, but by the way, what's your idea? Wrong one again. Why 1 in male and 2 in female? Not always, but usually like this. Any idea?
Also speculation time. It's a very good point. It is very striking, right? So I mean, one could argue from the perspective, does this just reflect the B chromosome history? So if the scenario is correct that you start as a B chromosome, you rely on, at least in birds, you would rely on inheritance through female drive. So in female meiosis is the avenue. So as a GRC, you don't care about the male side. It doesn't matter. Why you would then necessarily have just one GRC in males is tricky. So does it just mean there is selection to have 2 GRCs in females, or selection to have just 1 in males? I guess it's a bit tricky to say at this point. And so here's another speculative avenue I would just like to emphasize, which is that if you think about it this way, this GRC system is kind of behaves like a sex chromosome in terms of being, in the germline at least, being X0. So right, in males basically you have one GRC, and females you have 2. So that's the X0 XX system. So if you think of it this way, that would be basically kind of on top of a ZW system. So what if, still speculative, so waving my hands, what if that is kind of some way to counter some of the negative effects of having a ZW system? For example, adjusting for dosage effects, for example, related to the toxic Z effect also then from that perspective. So again, this is completely speculative, but it could just maybe reflect that there's multiple different interests within the genome, both regarding to sex chromosomes as well as germline versus soma. That could just be, once you have this vehicle, this is what you do then, what you can do just because you can. Basically.
Amazing. So you're just saying that GRC is replacing ZW for X0, is it?
I wouldn't say it's replacing it, but maybe it's an additional.
All songbirds.
It's an additional sex chromosome system, maybe, if we call it this way, on top of the ZW. Again, speculative. But what if it helps minimizing some of the negative effects of the ZW system, or at least like some of the limitations of a ZW, right? Because for ZW system, of course, it's easier, so to say, to have female beneficial mutations on the W, but on the Z you have of course effects at both sexes, unless you have dosage differences. So from that perspective, having a XO system is also, of course, helping you with some male beneficial things as well. But again, it's completely speculative, and I'm very happy to be very wrong with this point.
Absolutely. Okay, thank you very much. Any more questions, Nikolai Borisovich?
Maybe if you have time for speculation, maybe I can ask one more question. You see, if we will find some new mechanism for everything, for example, for elimination of GRC, usually that means that this mechanism exists a lot of time already, and it was developed for quite different purpose. Yeah, and do you have any ideas about what kind of purpose may be had this mechanism for elimination of large DNA regions earlier?
Yeah, this is a very good question, Nikolai. So I personally, I would find this easier to explain if we had something just like a region in the genome, like you delete site-specific deletion or site-specific recombination in one part of the chromosome, so you delete that. Let's say this is something that's, for example, done in ciliates and also in lampreys, for example. They remove parts of specific chromosomes predictably. And myself with a transposon background, I see lots of avenues or potential for domesticated transposable elements for doing those site-specific rearrangements. Also, like in our own immune systems, for example, with the VDJ recombination. This is something that happens in our own bodies as well, like the somatic recombination, all guided by site-specific mechanisms. So maybe it's just reflecting my naivety, but I find it very difficult to kind of assume you can mark somehow an entire chromosome if we assume it mostly consists of paralogs to other chromosomes. So, you know, but in terms of sequence homology, there should be homology to most regions in the rest of the genome. So how can you then mark that unless there's some other epigenetic mechanism of doing so? So I would rather suspect there is something related to the centromere structure and the associated heterochromatin there, or something related in general to specific histone marks across this chromosome. But then also there, it would be still, I would find it difficult to assume how that would be, how the organism would still be able to differentiate between the A chromosomes and the GRC if there is so high sequence homology. That said, maybe there are regions that are very different, and we just haven't found them yet for the puzzle metaphor reasons, for the limitations in sequencing methods. And we're starting to look at long reads in this context, and maybe then we are going to find those regions, and it might be a very simple explanation. But yeah, I don't know if any of you have a suggestion for what exactly might be marking this. Mechanistically, I would just say, what if it is a similar mechanism as in the elimination of the GRC during spermatogenesis? And I don't know if it's actually understood there how the GRC there is found or marked as being dumped soon, basically.
Well, it's look like it marked by centromeric sequence at least, because in males it shows very heavy labeling with anti-centromeric antibodies, but not in female meiosis.
I see. Okay.
Also in somatic cells, but we have to see. Yeah.
Nice.
Okay. Any more questions? No.
Thank you very much,
Alex.
Thank you so much.
Борис, у тебя есть вопрос еще? Нет.
Yep, I just say a lot of pleasure for me. Thank you.
Yeah. Thank you very much,
as well. Thank you so much
for most beautiful lecture, most inspiring and most close to our heart. Okay, so thank you very much. Thank you for all participants. Our next lecture will be on the next Wednesday. It will be Ilya Akberdin about, guess what? Epidemiology of COVID-19. Take care of yourself. Okay, thanks a lot. Thanks a lot, Alex. Thanks a lot, all.